Scientists have engineered bacterial expression of an anti-HIV compound, showing promise for ramping up its production. Welcome to Microbial Minutes, ASM's update on what's hot in the microbial sciences. This is the Better Living with Biology edition. I'm Julie Wolf, Science Communication Specialist here at ASM. And today we're going to be highlighting this recent paper from Science magazine that talks about Islatravir, a promising compound whose structure is shown right here on the slide. This compound works as a nucleoside analog. It is incorporated into the growing genomic contents of the virus, and it changes the genomic content structure, as you can see in the schematic here. So there's delayed chain termination rather than terminating the chain immediately. Islatravir shows a lot of promise in anti-HIV medication, which has already undergone several rounds of different clinical trials, and in fact was highlighted in the New York Times earlier this year. And I just want to point out this passage from the New York Times, where the CMO, the Chief Medical Officer of Merck, one of the companies involved, highlights some of the important attributes of this compound. It is much more potent, 10 times more potent than previous HIV drugs, meaning that you would need less of the compound to be effective, and this would also decrease the chance of bad side effects when one takes the drug. It also has a longer half-life, meaning that it will stay in the body of the patient for a longer amount of time. That would require fewer times of administering the drug, so one would only have to take or have this administered less frequently at any rate. Until now, Islatravir has been chemically synthesized in a multi-step process that has many different steps and requires things such as purification, and this can really slow down the process of generating a compound that is safe to inject or to serve to different patients. And so now, two companies, one of them being Merck, have announced the new bioengineering method to make scaling up of this Islatravir synthesis more efficient. Next page. So the enzymatic method for producing Islatravir was inspired by the bacterial nucleoside salvage pathway, and that's shown in the gray box here. There are three key enzymes that are needed, the DERA, the PPM, and PNP, which are shown here. And those were genes which were engineered based on either E. coli, although not the native E. coli genes. They were engineered to be more optimized for this particular production purpose, or Shigella halifaxensis. The team needed this particular reaction to run in the reverse from that salvage pathway, which is possible. Many biochemistry students out there may know or remember that reactions can often run in multiple directions, and the equilibrium of product to substrate is what favors one direction versus the other. And so in order to favor the reaction direction that they wanted it to run in, the scientists would remove one of the products that was made along with Islatravir, which is phosphate. And they did that by including a gene called a sucrose phosphorylase enzyme, which would use that phosphate group in the phosphorylation reaction. And this would remove the free phosphate and move the equilibrium, the reaction equilibrium, toward the product formation and increase their yield of Islatravir. They also needed to add different genes to generate the precursors. And so to do that, they screened a number of different bacterial kinase enzymes, finding that the E. coli pan-K gene was naturally the most efficient, but this was still pretty low efficiency at about a 1% yield. And so to increase the efficiency of this particular reaction, which they were running, they used directed evolution. They underwent 12 rounds of different selection in order to improve that precursor yield. This was then shunted into the engineered genes from the nucleoside salvage pathway. The resulting pathway, all engineered inside of E. coli as the chassis, I should mention, allows a one-step production of Islatravir. Now, on the next slide, we'll see that this was announced. Islatravir results from the clinical trial were announced earlier this year. And so, of course, Merck and the company with which it worked to produce this pathway, Codexis, have vested interest in this continuing on within studies. The company Codexis is very interesting. They are a protein optimizing company, and they've worked with Merck on a number of different projects for many different years. And the collaboration may result in this more efficient productive of a promising effective antiretroviral drug. We'll link to the stories that mention the Islatravir, as well as, of course, the original research down below. I'm also going to link to a story recently published on ASM that talks about the challenges of HIV testing, because, of course, HIV testing is the first step in getting properly treated. Today, we've heard about the creative synthesis process involved in bioengineering that is now going to be able to produce a chemically complex drug. For more updates, be sure to subscribe to ASM's YouTube channel and ring the bell for notifications every time we have a Microbial Minutes update. I'd like to thank you for listening and thank Ray Ortega for production. I'm Julie Wolf, and I'll speak with you next time on Microbial Minutes.